We'll be demonstrating various dimensionality reduction techniques using the MINS dataset, which consists of handwritten digit images. We will cover techniques such as PCA, TSNE, UMAP, ISOMAP, and autoencoders and compare the results. The MINS dataset has 70,000 grayscale images of digits from 0 to 9, each represented as a flattened vector of 784 features. Dimensionality reduction helps visualize and simplify this high dimensional data. First, we load the MINS dataset using the fetch open ML. Here, X_MINS contains the image data and Y_MINS contains the digit labels. Here is a sample digit from the dataset. First, we perform randomized PCA, a variant of PCA that is faster for large dataset. It reduces the data to 2 dimensions and we visualize the digits in this reduced space. As we can see, the digits cluster together, but there is some overlap due to linear nature of PCA. Next, we apply kernel PCA, which uses kernels like RBF to capture nonlinear patterns. This helps better separate the digit clusters. Notice that the kernel PCA separates the clusters slightly better. TSNE is a popular technique for visualizing high-dimensional data in 2D. Here I use a smaller subset of the data for faster processing. TSNE creates well-separated clusters, but it can be computationally expensive and sensitive to hyperparameters like perplexity. UMAP is another technique that excels in creating meaningful 2D representations. It's faster than TSNE and creates clearer clusters. Tools like pair code or understanding UMAP can help create interactive visualizations. ISOMAP, LLE, and MDS focus on preserving the relationships between data points. While LLE struggles with dense data, ISOMAP performs slightly better here. MDS tries to preserve distances, which leads to overlapping clusters. Finally, I implemented an autoencoder, a neural network designed to learn compressed representations of data. The autoencoder's 2D representation captures the structure of data effectively, similar to UMAP or TSNE. To summarize, TSNE and UMAP provide the clearest clusters, while PC and its variants were faster but less effective for this non-linear data. Reduction techniques on iris dataset, that is a tabular dataset. We will use methods such as PCA, TSNE, UMAP, ISOMAP, and autoencoders and analyze their impact on the dataset. The iris dataset contains 150 sample of flowers with 4 features: sepal length, sepal width, petal length, and petal width. Each sample belongs to one of the 3 species. First, we load the dataset using load iris from sklearn. X iris contains the features and Y iris contains the species labels. We aim to reduce these 4 features to 2 for visualization. We start with randomized PCA. It reduces the data to 2 components while preserving as much variance as possible. The plot shows the 3 species from overlapping clusters, indicating PCA is not fully separating them. Next, we use kernel PCA with a polynomial kernel to capture nonlinear relationships in the data. The plot shows how kernel PCA creates more defined clusters compared to linear PCA. Using TSNE, we reduce the data to 2D for visualization. TSNE separates the species better than PCA, but it's computationally more expensive. UMAP is another technique that creates well-separated clusters in a 2D space. UMAP is faster than TSNE and produces similar results, making it ideal for larger dataset. ISOMAP, LLE, and MDS try to preserve distances between data points. While ISOMAP and LLE focuses on manifold structures, MDS preserves pairwise distances, leading to overlapping clusters. Finally, I implemented an autoencoder for dimensionality reduction. The encoder learns a compressed 2D representation of the data. The autoencoder cluster are similar to UMAP, showing its effectiveness in capturing the underlying structure. To summarize, UMAP and TSNE provided the best separation of species in the iris dataset, while PC and its variants were less effective. Reduction. We will be studying PCA on a dataset of light sheet imaging. The dataset is generated by studying the movement of larval zebrafish, an animal that is specially useful in neuroscience because it is transparent, making it possible to record activity over its entire brain. In the visualizations below, we specify the mean of each dimension to be 50 and the variance along each dimension to be 1. We will explore two different values for the covariance 0 and 0.9. When the covariance is 0, the two dimensions are uncorrelated and hence the data looks spherical. In contrast, when the variance is zero point 0.9, the two dimensions are strongly correlated and thus the data is non-spherical. Now, PCA can be interpreted as identifying the direction along which the data vary the most. In the first step of PCA, we center our data. Working with the correlated dataset, we compute the mean of each feature, that is column in the dataset. The correlated data is an RDD of NumPy arrays. This allows us to perform certain operations more succinctly. Now we compute the sample covariance matrix. We To compute this matrix, we compute the outer product of each data point, add together these outer products, and divide the number of data points. The data are two-dimensional, so the resulting covariance matrix should be a 2 cross 2 matrix. Next, we use the expressions above to write a function to compute the sample covariance matrix for an arbitrary data RDD. After computing the sample covariance matrix, we use it to find directions of maximal variance in the data. We perform eigen decomposition of this matrix to find its eigenvalues and eigenvectors. Eigenvectors returned by I appear in the column and not the rows. The first eigenvector of I underscore x would be found in the first column and could be accessed using I underscore x index 0. We computed the top principal component for two-dimensional non-spherical data set. Now we use this principal component to derive a one-dimensional representation for the original data. Now we have all the required functions to write a general PCA function. Instead of working with just the top principal component, our function will compute the top k principal components and principal cores for a given data set. Next, we use the PCA function we developed to find the top two principal components of the spherical data we created in visualization. The original data is plotted as before, however, the one-dimensional reconstruction is plotted in green on top of the original data, and vectors representing the two principal components are shown as dotted lines. After working with two-dimensional data, now we work with three-dimensional data. We include the 2D plane that corresponds to the two principal components. Note that the darker blue corresponds to the points with higher values for the third dimension. Now we quantify the variance. We compute the fraction of retained variance by the top principal components.